Okay, my time just jumped, so let's get the show on the road. So welcome everybody to the keynote of the Cell and Developmental Biology subgroup. I'm Andreas Buttenschön from UBC and the outgoing chairman of the subgroup, and I will chair the session. So for some logistic information as before, please post your questions in the Q&A or the chat, and then we'll select a selection of questions at the end to pose to Adam. So now to the exciting part for our keynote today. It's my pleasure to introduce Adam Martin, who currently leads the Lab of Morphogenesis at MIT. And in particular, his group is interested in elucidating the mechanisms by which tissues get their shape. And for this purpose, he develops tools to visualize and quantify the dynamics of molecules, cells, and tissues. And with these tools, his group in particularly focuses on the impact of mechanical forces on cell behavior and how these shape the tissues. And with that, Adam, I hand it over to you and let you introduce everybody to tissue origami.
All right, great. Well, thank you, Andreas. And I'd like to thank the organizers for the opportunity to come and talk to you today. This is actually my first SMB meeting. And so it's been very enjoyable so far. And I want to start my talk just by acknowledging that I am trained as a biologist, and specifically a cell biologist, and the math that I'm going to talk about later in my talk has been the result of a really excellent collaboration with a colleague of mine at MIT, Jorn Dunkel, who's in the math department, and several of his trainees, specifically Norbert Stoop and Pearson Miller, have really contributed to providing us with insight into our process through mechanical modeling and simulation. The work that is done in my lab is the work of Hanna Jevic, who I'll focus on at the end. And this has been using math approaches to quantify network organization of connections in a tissue. So those are really, this work is wouldn't be possible without all of these people. And Hannah is trained as a physicist, I should mention. So the question that has really intrigued me in my lab for many years now is how during development does an organism go from essentially a puddle of cells to an organism with organs of intricate shapes? And so I've chosen here just two developing organ systems where I think it's clear how the shapes of these organs are of critical importance to their function, okay? On the left is a developing lung in a mouse, and on the right over here is a chick gut, and you can see the tube coiling around for the gut here. Now, both of these organs are composed in large part by a type of tissue that's known as an epithelium. And what an epithelium is it's essentially a tissue that's a bunch of cells that are packed together, and they often acquire this columnar morphology, shown here. And these epithelia also typically have a polarity to them, an inside-out polarity, which is known as apical basal. And there are different proteins that differentially localize apically versus basally. And one of these proteins is a complex known as the adherens junctions. And the adherens junctions is a complex that physically links cells together, as you can see molecularly here. It's like a Velcro between cells, and this typically localizes towards the apical side of an epithelial cell. Now, during development, epithelia undergo all sorts of different morphological changes in order to sculpt tissues into their final shape. And one that I think is very dramatic is when a two-dimensional epithelium bends and folds to generate three-dimensional shape. And this happens fairly ubiquitously during development. I'll just show a few examples. Here's a tissue that bent into this characteristic cup shape, which is a developing eye, in this case in zebrafish. And here's another example of a mouse neural tube that's folding and zipping up along this seam here. It's just looping over and over again. And this is important because this will give rise to the vertebrate brain and spinal cord. And defects in this folding process result in some common human birth defects. So it's really important that we understand mechanistically how force is generated to drive these processes. And luckily, because we want a really mechanistic understanding of the forces that are generated during this process. Luckily for us, epithelial folding also happens in some really powerful model systems to study development, such as the fruit fly Drosophila. And what's shown here is a Drosophila embryo. It's an ellipsoid-shaped embryo that's about 0.5 millimeter long. And this is about 2 hours and 45 minutes into development. And you can see at this developmental time point, the surface of the embryo is relatively smooth. There is a cobblestone-like appearance to the embryo, and each cobblestone here is an individual cell. Now, what happens at 2 hours and 50 minutes in development is a population of about 1,000 cells on the ventral side of this embryo will invaginate and fold inwards to generate a crease along the ventral midline, which you see here. Okay, and this is known as the ventral furrow. Now, to show you another viewpoint of this, you can imagine this embryo being a loaf of bread. You can take a knife and cut it in half and then look at it from the end on to see the loaf or the embryo in cross-section. And then looking at this viewpoint, here you see the cell membranes labeled so that you can see individual cells. And you see embryos at different stages of this folding process. And what I want to point out is if you focus on the cells highlighted in orange, you can see before the process starts, the cell is on the outside of the embryo. And at the end of this process, these cells are on the inside of the embryo, where they will eventually form a new tissue layer. Okay, so this process is essentially taking cells initially located on the outside and putting them on the inside. You also see a dramatic shape change in these cells over this time course. And you see that cells, while they start as being columnar, they constrict their outer or apical surface in this case, and that causes them to become wedge-shaped or bottle-shaped. And it's this collective shape change known as apical constriction which leads to the bending and ultimately the invagination of these cells into the interior of the embryo. And here's just a movie showing you these cells invaginating. You can see the dark holes are the nuclei of the cells, and you can see how these ventral cells move inwards. Now, I think a useful analogy in thinking about this problem is origami, right? Origami allows you to take a sheet of something, being a piece of paper, and by making creases in the paper, it allows you to generate a three-dimensional form. Okay, so if you start with this flat sheet of paper and you bend it in a stereotypic way, you can generate this origami crane. Okay, and you all know that this can be a very reproducible way to generate three-dimensional shape, as illustrated by this flock of origami cranes. Okay, and now to take this analogy a step further, I want you to think about sort of the steps that go into doing origami. First of all, you might start by deciding where on this sheet of paper you're going to put the folds or the creases. And then to start the process of generating three-dimensional form, you would initiate a crease or a fold by generating a local force, such as a pinch. And then in order to finalize the crease, you have to propagate force along the length of the sheet, okay? And this will essentially be the organization of my talk. I'm going to take you through the process of tissue folding from the molecular level at the nanometer scale all the way up to the tissue level, which is on a millimeter scale, okay? So I'll start by telling you about the genes and the molecular motors that are involved in deciding where on the embryo this fold is going to go. I'm then going to tell you about how cells are organized to generate force in order to start the fold. And then I'm going to tell you about how force is propagated across the entire sheet in order to generate a crease, okay? So these will be the three parts of the talk. So I'll start with what I feel is the hero of the talk, which is a molecular motor. The motor is a type 2 myosin, which I'll just refer to as myosin. It's the same class of myosin motor that is found in your muscles and leads to muscle contraction. But it also has a non-muscle flavor to it. And this myosin assembles into oligomers, which form bipolar filaments, where you have the heads of the motor sticking off the ends of the filament in different directions. And if we focus in on an individual pair of heads, I'll show you what these heads do. These heads, motor heads, use the chemical energy of ATP hydrolysis to undergo conformational changes that move this motor along an actin filament track shown here, and it moves unidirectionally towards the plus end of the actin filament, okay? So that's myosin. Now, because generating force is an incredibly powerful thing for a cell to do, it has to be highly regulated, right? If it's done inappropriately, that can lead to disease, it can lead to all sorts of problems. And so in non-muscle cells, this is regulated by a small GTPase known as rho. And this rho GTPase coordinately activates both the myosin motor and the assembly of these actin filament tracks. And I'm going to focus on this branch of the pathway. The way it activates the motor is through its effector kinase called rho kinase, which phosphorylates myosin, leading to conformational changes that allow the myosin to assemble into these bipolar filaments. Now together, actin and myosin composes a contractile system where myosin mini filaments, through the activity of the motor heads, can take antiparallel actin filaments and slide them together, generating a contractile force. So what does this look like in this problem of tissue folding that I introduced? So I'm going to show you the Drosophila embryo again, and I showed you earlier this cross-section image where you can see it starting to fold. And you can see the cells, how they're constricting at the surface. And now I'm going to show you a different viewpoint. There will still be a cross-sectional view over here, but I'm also going to show you a bird's eye view looking down on the surface of the tissue right here. And these embryos are expressing a marker that labels the plasma membrane of the cells. And that's shown in magenta. And I focused on a subapical slice that allows you to see the outlines of individual cells. So each one of these sort of hexagonal or units here is an individual cell. This embryo is also expressing a labeled myosin. And for myosin, I'm going to show you a projection that includes the surface of the embryo, which is where the myosin is assembling. And if you look at the cross-section, you'll see how myosin is really restricted to the very surface of the embryo. So I'm going to play the movie right now. You're going to see myosin accumulate here. Now the cells with myosin disappear into the inside and the surrounding cells zip up. Okay, I'll show that one more time. There, the surrounding cells come together and seal up. Now I'm just going to stop the movie right here, and you can see how myosin is enriched in the cells that are making this furrow. And again, I point out myosin is highly enriched at the surface of the embryo. So for the remainder of my talk, when I show you something in green, I'm showing you a component of the cytoskeleton, such as actin or myosin, or I'm showing you a regulator of the cytoskeleton like rho kinase. When I show you something in magenta, it's going to be something that's subapical and showing you the outlines of the cells. So now to get to the question of how is it that the embryo decides where to put this fold. And the work I'm going to describe today is a compendium of decades of genetics that's been done in the field. And the pioneers in this field have really been Eric Wishaus, Maria Leptin, and Norbert Perimon, just to name a few. They've done the really hard genetics to sort of understand how the embryo decides where to put this fold. And all the work I'm going to tell you about from my own lab has really been sort of done on the shoulders of these folks here in their labs. So what these groups found is that there are master regulators at the top of a cascade, and these, I'll start with these two transcription factors, snail and twist, which are expressed specifically in these cells that are going to invaginate. So here's a snail here in green, and it's labeling the nuclei of the cells that will eventually go inside the embryo. And these transcription factors regulate the expression of other genes, and two of these genes encode for signaling proteins that then signal to the rho GTPase, shown here, and that then activates actomyosin contractility. Okay, so this is the genetics of it, but as Robert Insall said yesterday, the genetics only gives you a limited understanding of the actual mechanism. And I guess the picture that arises from genetics is the following, that you have a cell that would be expressing snail and twist, and it leads to the expression of a signaling molecule that's secreted apically, and that apical signal would then lead to the activation of rho on the apical side of the cell, and that activation would then lead to actomyosin activation and apical constriction. Okay, this is the model from genetics. Okay, makes a lot of sense. So what a postdoc in my lab, or former postdoc in my lab, and I did, Frank Myosin, is he was interested in mechanistically how these cells constrict. Both Frank and I are cell biologists. We want to see the dynamics of this process. We want to see how things are organized within cells. And consistent with the model, what we did was we started with rho kinase, which is the signaling protein just upstream of myosin. And consistent with the genetic model, you can see here rho kinase is apically enriched in these cells that are undergoing apical constriction. Now the surprise came when Frank decided to look at a gene that was upstream of rho kinase, and we looked at the transcription factor twist. We looked in a twist mutant, which based on the model should disrupt rho kinase activation. But what we found is that rho kinase continued to apically localize even in the absence of this master regulator, which was surprising. Now, Frank looked more carefully and discovered something interesting. So I just want to point out the twist mutant. In the twist mutant, cells do not constrict, and the tissue does not fold, even though you have this apical rho kinase. So Frank looked more carefully and found that rho kinase is not just uniformly localized across the apex of these cells. Now this view is looking bird's eye view down at the surface of the tissue, similar to what I showed before. And what you see is that there's a patch of rho kinase in the middle of the apical domain of each of these cells. And what we found is that in the twist mutant, the polarity of this rho kinase is inverted so that the rho kinase spots are instead localized to the junction rather than the center of the apex. Okay, so this provided a different picture of this problem, where now it appears there's a signaling center of rho kinase that's present in the middle of the apex of these cells. Okay, so we wanted to understand, why is it that rho kinase and myosin has to be in the middle of these cells? And this got us thinking about muscle, because I'm a cell biologist. I love thinking about muscle. And in muscle fibers are made up of a repeated contractile unit known as a sarcomere. Shown here is an electron micrograph of one sarcomere, goes from here to here. Okay, and a key feature of this sarcomere is that myosin localizes to the middle of this unit. You can see the heads of the myosin are these dark bands here and here. And so the other key feature is that there are anti-parallel arrays of actin filaments with the plus ends of the actin filaments facing outwards, okay? And because myosin is the heads are going to walk towards the plus end, when myosin gets activated here, it slides these anti-parallel arrays together and shortens the contractile unit. So Jonathan Corvus, a former graduate student in my lab, decided that for us to understand the mechanism of apical constriction, we needed to know the polarity of the underlying actin network. And so what was previously thought about this process is that you just have a random network of actin filaments with no polarity. But another possibility is that there is some polarity to this network, and we wanted to know if there was polarity and what that polarity was. So the challenge here is that if you just label the sides of the actin filaments and label all the actin, you get this really complicated meshwork, but you can't see which end of the actin filament is where, okay? So this tells you there's a meshwork, but it doesn't tell you whether there's a polarity. And so what Jonathan did was to devise ways to label one end of the filament and the other in order to determine whether there's a polarity to the network. And to label the plus ends of the actin filaments, what he did was to purify actin monomers. Remember, actin is a biopolymer that assembles into filaments, and it assembles preferentially at the plus end, especially when actin is in complex with profilin. So what Jonathan did was to inject labeled actin subunits with profilin and look to see where these incorporate. And what he saw is that they preferentially incorporate at the junctions of the cells. And in a reciprocal experiment, he labeled the minus ends of the actin filaments using a minus end binding protein tropomodulin. And what he found is that tropomodulin localizes to the center of these cells, suggesting a polarity where you have plus ends enriched at the periphery of the apex and minus ends enriched at the centroid of the apex, and taken together with the signaling center I showed you, where myosin is getting activated preferentially in the center, what you have is a system where along all radial angles around the centroid of the cell, you're going to have anti-parallel networks of actin filaments and the centrally localized myosin that could be bridging them, which would lead to myosin reeling in these actin filaments and pulling them towards the center to generate a constriction. So this organization is remarkably like a sarcomere, except it's radially arranged around the signaling center. Now, if we take a step outwards and look at this on a more tissue scale, there's, I think, a really interesting patterning problem here. And this is a problem that we're still working on to try to figure out how this pattern is established. But this is an image of rho kinase. And what you see is this almost looks like a cheetah's spots, right? You have a polka dot pattern where you have patches of rho kinase that are repeated sort of over a spatial scale. But in this case, you know, you might be familiar with Turing patterns. We don't think this is a Turing pattern because the spacing of these spots is really based on the organization of the cell layer. So here are the outlines of the cells. And what you see is that each cell has a spot of rho kinase in the middle. There are other rho kinase puncta in a finer scale organization to it. But in general, each cell has a signaling center of rho kinase, which is highlighted here in yellow. And what that means, because these are contractile units, what it means is you can think of this tissue as assembling a two-dimensional grid of contractile units, which are all connected to each other through adherence junctions, which physically link the cells to each other, which are shown here in blue. And they're all pulling against each other in the tissue. And that pulling is going to generate tension, as I'm going to show you in just a minute. Okay, but I just want to set the stage for how you should think about what these cells, how the cells are organized, and how they're all cooperating together to pull against each other to fold this tissue. Okay, so now I'm going to switch gears, and I'm going to tell you about the last story, which is how are forces transmitted across this sheet, and how does this impact the overall tissue shape? And I'm going to tell you basically a synthesis of work from a number of different people that's been done in my lab over the years. I'm going to start with work done by a former graduate student in my lab, Claudia Vasquez, who did work sort of optimizing our inference methods to measure forces in these sheets, and also showed that these forces are dependent on myosin. And then I'll tell you about work done by Soline Chenet, who did experiments to alter the pattern of forces in the tissue to show us what morphologically happens. And then I'll tell you also about work done by Hanna Jevic, who developed quantitative approaches to map how forces are transmitted through a tissue, and also worked with the Dunkel lab to show how that impacts tissue shape. So first I want to tell you about how we infer forces in tissues. And we use a method that conceptually is very simple. So if you consider this elastic band, if this elastic band is under tension and you cut it in the middle here, it should recoil. Okay, so here's a simulation by Claudia, where she showed how when she cuts it, the ends of the band recoil. And if you measured the velocity of the ends of this band, you could get some sort of displacement curve. And this initial recoil velocity is proportional to the amount of tension that was in that material. And there are a lot of caveats to this experiment, but to first approximation, this is a pretty good way to compare relative amounts of tension in the tissue. I just want to point out that this method was pioneered by Shane Hudson in collaboration with Glenn Edwards and Dan Keyhart. And Shane gave a talk at the meeting yesterday. And actually, this is one of my favorite papers, science paper from 2003. And this honestly is what got me into this field of tissue folding. So what's going on during the folding process? I'm going to show you an experiment that Claudia Vasquez did. What's shown here is the surface of the tissue in the ventral domain. And you're seeing myosin labeled here. You can see some patches of myosin, but you can see it's also forming a more complicated network. And what Claudia is going to do is take an infrared laser and irradiate the cytoskeleton along this yellow dotted line and that will basically burn it and cut a hole in it, and you can see the resulting myosin structures recoil away. There's the cut, boom, you can see the recoil, okay? We'll keep that looping a couple times so you can see it again. There's the cut. So we could then track these structures and measure recoil velocity to infer the relative amounts of tension in this tissue. And what Céline Chanet did was to do this experiment in a couple different ways. She made incisions in this direction and measured recoil along the long axis of the embryo, which is the anterior-posterior axis. And she got recoil velocities of this magnitude. And then she did the orthogonal experiment where she made cuts that were aligned with this axis and measured recoil along the ventral lateral direction and saw that there was a twofold lower recoil velocity, okay? So this suggested that there was anisotropic tension that's generated in this tissue during the folding process, where the tension is oriented along the length of the furrow and the long axis of the embryo. Now, to see if there is other types of anisotropies or other evidence for anisotropic force generation in this tissue, we also looked at the organization of the actomyosin cytoskeleton. And what we found is that if you look at myosin, you can see here, this is a labeled myosin. And again, you can see that there are sort of intent myosin nodes in the middle of the cell apex, but you can see that these nodes often connect to each other through myosin fibers, and these are actomyosin fibers. So a molecular interpretation for what these fibers represent is these fibers are reaching out from this central myosin node and connecting to adherence junction structures that are providing a mechanical anchor point between cells so that essentially myosin fibers can connect between cells and the cells can pull against each other. Okay, so the myosin, the cytoskeleton is forming a tissue-wide or supracellular meshwork across hundreds of cells in the tissue. And again, I want to show you, this is a little bit more zoomed out. You see the myosin network behind me and in my Zoom background, but now you're also seeing it here in green on the slides. And you can see how myosin is forming this fibrous network of connections, which is rather complicated. And if you take a couple individual examples, look at this cell here. You see how the fiber is aligned with the anterior-posterior axis. Okay, so that's just an individual case, and I'm going to show you a more rigorous analysis in just a minute. But you see how the fiber is oriented. And that's the axis of the tension. So Hannah Yevik in my lab decided or got an idea from Jorn Dunkel, who suggested we use this algorithm known as disperse, which is an algorithm that was used in cosmological research to basically map the filamentous structure of dark matter in the universe. But it's a technique that's topological and based on Morse-Smale theory. And it allows us to take a sort of image of myosin, and it identifies persistent topological features in this image, such as local maxima, and also edges that connect those local maxima, which the maxima, again, are sort of these local signaling centers inside each individual cell. And the edges are these myosin fibers that are connecting between cells. So this allowed us to represent this myosin network in a framework that's described by this node and edge description, which allowed us to simplify this greatly. And what it allowed Hannah to do is really quantitatively analyze this network and analyze properties such as the orientation of these edges that connect distinct nodes. And what she found when she measured orientation is that there's actually a connectivity bias mechanically between the cells in this tissue, where there are more anterior-posterior connections than there are ventral lateral or ventral dorsal connections. So not only are there aligned fibers, but there's really more connectivity along the axis of higher tension than there is along the other axis. This type of analysis also allowed her to analyze properties of these connections, such as their straightness. And what Hannah found is that anterior-posterior connections are actually straighter than the ventrolateral connections, again, consistent with the idea that there's higher tension along the anterior-posterior axis of this tissue. So next we wanted to try to get insight into why the embryo has this anisotropic tension. Essentially, what's the function of this oriented tension and oriented cytoskeletal connections in terms of generating a proper tissue shape? And to get at that, we collaborated with the Dunkel lab, and we collaborated with a number of members of this lab, including Pearson Miller, Norbert Stoop, and Aidan Furrow. And all our interactions really sort of helped us get better insight into why you need directed tension in the system. And I'm going to start with just the base model that they created of basically this folding process. And this is a very simple model, but I'll show you how it captures some of the essential features of the system. The model is a Coiter shell model, and basically they modeled an elastic shell that's roughly the same shape as a Drosophila embryo. And they basically, the way the model works is you have this elastic shell, and the middle surface has a reference shape, and there's an energy penalty for sort of deviation from this shape. And that allows the system to sort of evolve into different shapes as you change the properties of this reference shape. And so what contributes to the energy are stretching deformations, bending deformations, and also shear stress. And so to model different patterns of contractility, what the Dunkel lab did, which I thought was quite ingenious, is to do something relatively simple, which is if you consider this toy model here, you have a top surface, A+, and a bottom surface, A-, and they inferred for a given level of myosin contractility on the top surface, because these surfaces are connected, it's going to lead to a specific preferred curvature for a certain amount of contractility. And you can think of this kind of like a bimetallic strip, where if one of the metals expands relative to the other, because the metals are connected, it generates a curvature, okay? So then we could model different patterns of contractility in this Coiter shell model, because by basically imposing a different preferred curvature based on the pattern of myosin here. And so first what we did was to try to figure out what the pattern of contractility is for a normal wild type embryo. And I've told you how there's a domain of cells that express these transcription factors. But what we discovered is that the level of contractility in this domain is not uniform, but is actually present in a gradient, with the highest level of contractility being right along the ventral midline and the levels dropping off as you go to the sides of the embryo. And so when the Dunkel lab did simulations with their model, given the wild type pattern of contractility shown here, what they found is that this is a cross-section through their simulation. This gradient in contractility leads to a very nice fold in their simulation. And what the Dunkel lab did is they also played with this pattern, and they made a prediction that if you expand the width of this domain, of this contractile domain, you wouldn't just get a deeper and deeper furrow, you actually get a transition from a fold to something that looks like a flattened depression. Okay? So this was a prediction of their model, and it's something we were able to test because we were able to develop an experimental perturbation which expands the ventral domain, shown here. We basically knock down and deplete an inhibitor of ventral cell fate, and you get more cells of ventral fate. So remember, in wild type, you have a gradient of contractility, and you get this nice sharp fold, shown here. When we expand the contractile domain, what happens is you get a morphology that's similar to what was predicted by the model, where you get a flattened depression, shown here. Okay, so this shows how this relatively simple model is able to capture essential features of the system, including this mutant phenotype. So just to give you an intuitive explanation for the reason for this, what we think the gradient does is that it provides a force imbalance. So if you're a cell in this domain, you're going to be pulling harder than your neighbors that are more on the side of the embryo, and that allows these cells to constrict better along this axis, which leads to higher curvature. Whereas if you consider the orthogonal axis, the long axis, what you have is a force balance and a giant tug of war between cells that just generates tension rather than generating efficient constriction. And what we think happens in the mutant is it converts sort of this situation more towards this situation. And we've shown that that's the case, where you actually generate higher tension along this axis in the mutant than you do in the wild type scenario, okay? So it's really the geometry of this contractile domain is contributing to the pattern of forces. So now we wanted to determine what is this supracellular network of actomyosin connections doing for tissue shape. And so what we did is we took the Dunkel Labs Coiter shell model, which we had working, and I showed captures some features that we observed. And we superimposed on the surface of this model a stiff network that topologically resembled the wild type network that we could quantify, okay? Because we inferred that if you have these fibrous elements that are under tension, they're tense and they're going to be stiffer relative to the surrounding tissue. And so in including this network of stiff elements, what we found is that if we make the network biased towards the anterior-posterior axis, like we see in a wild type embryo, it makes the folding faster than what you would see without this network. So what you're seeing here on the y-axis is the depth of the invagination, and you can see how the invagination here is shifted to the left, which is earlier in time. And what we saw if we generated an opposite type of network where the connections are biased towards the ventrolateral or dorsal ventral axis is that this actually slows down the folding, right? So if you don't have cells connecting in the right way, you can actually impede the fold, and that's important. Another thing I want to point out is this model, we explored what happens if you expand the contractile domain a little bit more. And sometimes what we saw in the model is that rather than getting a furrow that forms along the long axis, you would sometimes get this case where you get a transverse fold in the simulation. And so what we then did was we wanted to test whether experimentally, whether geometry affects the orientation of cytoskeletal structures. And what we did is to look at the organization of actomyosin in this knockdown where you have an expanded contractile domain. And I just want to remind you that in wild type, you have actomyosin fibers that are oriented with the axis of tension and are biased towards the anterior-posterior axis. That's this axis. When we expand the contractile domain and change the geometry, we totally change the organization of the cytoskeleton. And rather than getting oriented actomyosin fibers, we instead get an actomyosin ring in each cell. And when we analyze the tissue level network organization, you can see we lose the anterior-posterior bias to connectivity. Okay, so now we have in this mutant situation, we are disrupting the oriented cytoskeleton, and I've also mentioned to you, it also disrupts the oriented tension. And what we see about 50% of the time in this situation is, as predicted by the model, you get transverse folds forming with the wrong orientation. So what we think this oriented cytoskeleton and tension is doing for tissue morphology is that this network of oriented cytoskeletal fibers is suppressing misoriented folding events and is essentially channeling the tissue towards a furrow that's oriented in the same geometry as the genes that are expressed that are sort of the ones that are going to be driving the fate of these cells that invaginate. Okay, so you can think of it as the supracellular network is providing a frame around which the tissue shape is following. So just to summarize the take-home messages from this talk, I showed you that there's a sarcomere-like organization to the cytoskeleton that generates a local force. And finally, I showed you how a supracellular actomyosin network with biased connectivity promotes folding with proper orientation. And again, I'll just end by thanking the people who did the work. Frank Mason and Jonathan Korovas contributed the most to this idea of there being a radial sarcomere powering apical constriction. And then Claudia Vasquez, Soline Chanet, Natalie Here, and Hannah Yevik did the work showing how tissue level anisotropy and tissue level forces orient the proper, they orient the fold. And this was together with our tremendous collaborators, Jorn Dunkel, Norbert Stoop, Pearson Miller, and providing us with insight into this process. I also thank the FLY community for reagents and NIH and American Cancer Society for funding. So I'd also thank you for listening, and I'm happy to take any questions.
Yeah, thank you so very much, Adam, for this fantastic talk. There's a few questions that keep on rolling in on the chat. So let's start with a question from Alex Fletcher on the very first thing that you talked about. Do cell intercalations play a role at all in this process? He makes the astute observation that this might mean that it doesn't quite fit with your origami analogy.
So in the stages of folding that I've shown you, there is very little to no intercalation. So that's something that we have measured.
Excellent. Okay, I have another question by David Holloway, who asks, what localizes ROC in the twist mutant?
My answer to that is we're not entirely sure, and we're still looking at it. There are other factors other than Rho that can recruit ROC to places, and one of those is a protein called shroom. And the ROC localization that we see in the twist mutant is remarkably similar to shroom. localization. So I expect it might be shroom. There's another question about other organelles that are associated with these centers. And we just published a recent paper showing that there's also a microtubule organizing center that is a part of those centers. It's non-centrosomal, and but the microtubules are not required for forming those centers. The microtubules are doing something different. So if you're interested in that, I would take a look at our recent work on the microtubule cytoskeleton.
Yeah, there's also a question on, do forces generate during furrow formation feedback on the and do they feedback on the orientation of the actin fibers?
We think they do. And in fact, I think I have just another example here showing you how if you just change the shape of the embryo, you also change the pattern of forces. And that changes, again, the organization of the actomyosin. And we've also done this in other ways with laser ablation. If you make laser ablations and release tension along a certain direction, then you can either generate actomyosin rings or generate these fibers depending on how the overall forces are oriented. So I would say yes, we think that the forces in the tissue are sort of feeding back and leading to actomyosin alignment. And we actually collaborated with Lance Davidson to have an agent-based model of the cytoskeleton that also captures this phenomenon. There's another question here about clinical implications. I think it's still a little early for that, but I will say that the mouse neural tube does have a supracellular actomyosin network. And from the exome sequencing of human spina bifida cases, there are a number of cytoskeletal components that come up in those sequencing studies. So it's likely that this does have relevance to birth defects in humans.
There also seems to be a more pointed question here, so let me read it out.
Okay,
In
sure.
the experiment SPN27A RNA-I, it seems that the contractile domain was not only non-graded, but also broader. Does the width of the contractile domain contribute to the phenotype as well?
We think so based on the fact that if you cut within that uniform domain, it will sort of reorient the cytoskeleton. And so I guess my answer to that is yes, we do think that.
And from what I can see, that I think I covered them all now, or we covered them all now, as far as I can tell. Somebody very quickly yell into the chat if that's not the case. People are saying that they really enjoyed your talk now. Ah, there we go. Okay, there are some people that I have missed. Have you checked if there are any relations between this change of the shape and the chromatin structure of these cells?
We have not.
And then I guess, oh, I missed the last part of Roland. Was it possible to keep changing only the shape of the boundary of expression domain? Oh, I guess, was it possible to keep only changing the shape of the boundary of the expression domain? Yeah,
can we change only the boundary? We haven't figured out how to do that yet.
Okay. Oh, now the questions keep on rolling in. Can you see the chat by any chance? Otherwise, I'll keep reading them to you. Okay. So we have a question here. What's the correlation between the AM anisotropy and cell shape anisotropy?
Both are about twofold.
And then I guess we have time for one last one. How about active polar gel models where dynamics and organization of actin is modeled based on free energies and movement of cells as flow velocities? Do you feel such models can describe overall dynamics of the embryo in the future?
We haven't done that per se. In the model we did with Lance Davidson, we didn't model flow velocity of like surrounding cells, but we varied the strength of the attachment to a boundary. So I think it captures some of the features as to what you're suggesting this other approach could do, but it might be different.
Well, I think I caught them all now. So I would like to thank you very much on behalf of us all for accepting our invitation to speak and for giving us your time to give us this excellent seminar.
You're very welcome.
Thank you very much.
My kids didn't walk in. That's great.
That's fantastic. Well, I hope the flies in your basement are still alive.
Oh, they're good. They moved back to the lab.
Oh, excellent. There's some progress here then. Well, thank you so much, and I hope to meet you sometime in the future.
Yeah, hopefully we are able to meet in person at some point.
Yeah, that would be excellent.
All right, great. Take care. Have a good day.